Okay, so welcome back to this next video in which we are discussing Cys-Loop ligand-gated ion channels. Okay, so we're currently in the process of discussing examples of Cys-Loop ligand-gated ion channels, and we're in the process of going through the ones that are present in humans. So we discussed that the nicotinic acetylcholine receptors, the GABA A receptors, and the glycine receptors are all these large families of ligand-gated ion channels, and these three families are within this bigger superfamily of Cys-Loop ligand-gated ion channels. Okay, now what we're going to do is complete our discussion of the mammalian ones with the 5-HT3 receptors, and then we'll go on to discuss some non-mammalian ones, okay, which include the glutamate-gated chloride channel from worms, okay, and the Glick and Eunuch channels from prokaryotes. Okay, right. So firstly, let's discuss then the 5-HT3 receptors, okay, or the type 3 5-HT receptors. Okay, so the 5-HT3Rs. So firstly, let me discuss the ligand for these receptors. So the ligand for these receptors is 5-HT, okay, which stands for 5-hydroxytryptamine. Okay, so the H is for hydroxy, okay, and the T then is for tryptamine, so 5-hydroxytryptamine. Now, I'll show you this molecule in a moment, but firstly, let me just tell you that 5-hydroxytryptamine has another name, and this other name is probably more famous than 5-hydroxytryptamine. 5-hydroxytryptamine, if you like, is the more medically correct name. However, its more common name that people generally know is serotonin. Okay, so 5-hydroxytryptamine, 5-HT, serotonin, they all refer to the same thing. So this is the ligand for 5-HT3 receptors. Okay, now, serotonin has a huge number of receptors for it in the human brain. Okay, 5-HT3 receptors, type 3 5-HT receptors, these are the only ones which are ligand-gated ion channels, and they're specifically in this superfamily of Cys-Loop ligand-gated ion channels. Okay, all of the other receptors for 5-HT are not ligand-gated ion channels. Okay, they are G-protein coupled receptors. So the type 3 5-HT receptors is this family of receptors for serotonin that are ligand-gated ion channels, and specifically they're Cys-Loop ligand-gated ion channels. Okay, right, so let me show you the structure of 5-hydroxytryptamine. Since I've shown you the structure of all the other ligands, I think it would be unfair now not to show serotonin structure. So basically it's based on the structure of the amino acid tryptophan, okay, but what we're going to do is firstly remove the carboxylic acid group of the amino acid tryptophan to create tryptamine, and then we're going to stick an alcohol group on to create 5-hydroxytryptamine. So here's the amino group of the original tryptophan amino acid. Here's the alpha carbon with a hydrogen coming off it, but now the carboxylic acid group's gone, okay, because we don't want tryptophan, we want tryptamine, okay, so we've taken off the carboxylic acid group and replaced it with a hydrogen atom, and that's the conversion that turns tryptophan into tryptamine. Okay, now let's show the R group of tryptophan. Okay, so you have a methylene group here, and then off the methylene group you then have what's called an indole ring, which basically consists of a pyrrole ring attached to a benzene ring. So let me show this. Now I'm not going to show it in molecular formula, I'm going to suddenly move from showing the amino acid in molecular formula to showing it in skeletal formula, because the indole ring looks hideous if you show it in molecular formula, whereas it looks quite simple in skeletal formula. Okay, right, so firstly then, the indole ring consists of the pyrrole ring bound to the benzene ring. So let's firstly just show a pyrrole ring. So a pyrrole ring contains a nitrogen at the top with 4 carbons here, and it's got 2 double bonds, one there and another here, and then it will have a hydrogen coming off this nitrogen here. Okay, so that ring that I've shown you so far, okay, that 5-membered ring, that's called a pyrrole ring. Okay, and if we have the pure pyrrole ring, that would just consist of, chop this bit off here, and then you'd have hydrogens coming off these 4 carbons here, which we wouldn't show because we're showing a skeletal formula. Okay, right, so that's the pyrrole ring. Now to convert the pyrrole ring into the indole ring, which is the thing that you're going to have in the R group of tryptophan, okay, what you need to do is attach onto this pyrrole ring here a benzene ring, okay? So benzene is just this six-membered carbon ring where you have alternating double and single bonds between the carbons. So here's a double bond, here's a single bond, here's double, single. And then each of the carbons in a pure benzene ring would then have hydrogens coming off it. Obviously only these four here will have hydrogens coming off it in the indole ring, because these two are going to have the pyrrole ring components coming off. Okay, right, so that structure now is the indole ring. Okay, right, so we have now shown tryptamine. This is the structure of tryptamine, but we don't want tryptamine, we want 5-hydroxytryptamine. So let me just show you the numbering system for the components of this indole ring of tryptamine. Okay, so basically the nitrogen here is given the number 1. Okay, and then you just count round. This one's called number 2. This one's called number 3. Now, for some reason that I won't pretend to understand, chemists have decided that when you number the one that's in between two rings, you do not go on to another number. Instead, you call it the same number as the previous one, and then you put an A there. So this carbon here, which is in between the two rings, the pyrrole ring and the benzene ring, is not called number 4, but it's called number 3A instead. Okay? And then this one's called number 4. Okay, this one is then number 5, excellent. This one's number 6, this one's number 7, and then that one there is number 7A, because again, you don't go up to number 8, you call it 7, the one previously, and then just put an A there. Okay, right, so we now know where number 5 is on our indole ring. All we now have to do is stick an alcohol group on number 5 there. Okay, like so, and we've then got 5-hydroxytryptamine, 5-HT, which is the molecule serotonin. Okay, so there's the structure of serotonin then, which is the ligand for these 5-HT3 receptors. Okay, so serotonin is a very important neurotransmitter in the brain and also in the peripheral body. It is used peripherally as well, not just in the brain. Okay, and as I say, most of the receptors for serotonin in the brain are going to be G protein coupled receptors and therefore aren't going to cause changes in ionic currents across the cell membrane directly, at least. Okay, however, there is this family of serotonin receptors called the type 3 serotonin receptors, or 5-HT3 receptors, which are ligand-gated ion channels and are Cys-Loop ligand-gated ion channels. Okay, and when they open, they form a channel which is permeable to sodium and potassium ions. Okay, so cations. Right, and as I have described previously with the nicotinic acetylcholine receptors, the important one there is the sodium current, okay? Because when you open one of these channels in a cell which has a normal resting membrane potential, let's say maybe negative 65 millivolts, the electrical gradient is going to be in agreement with the concentration gradient for sodium, and therefore you're going to get big movement of sodium in, whereas the electrical gradient will be in opposition to the concentration gradient for potassium, and therefore you're not going to get that much movement of potassium at all. Okay, so the important current that you get when you open these channels is a sodium current in. Okay, and that's going to therefore depolarize the electrical potential difference across the cell membrane, making it more likely that that postsynaptic neuron is going to fire an action potential. So again, serotonin working through these 5-HT3 receptors is an excitatory neurotransmitter, basically. Okay, right, and again I'll stress that this is not a single receptor. The 5-HT3 receptor is not one type of receptor, it's a family of receptors. Okay, at present we only know 2 of them, okay, but there are probably more, and we'll come on to that later on when we discuss these in more detail after we discuss the structure of Cys-Loop ligand-gated ion channels and their gating mechanism in more detail. Right, okay, so now what I want to do is mention the non-human forms, the Cys-Loop ligand-gated ion channels that we found in other forms of life. Okay, so the first one I want to talk about is one that has been found in many different types of worms. Okay, and important for us is that it's present in certain parasitic worms. Okay, so worms then now. Okay, and in particular for us, we're going to be interested in helminths, which are just parasitic worms, okay, which are capable of causing disease in humans. Right, okay, so in worms and helminths, you have a cis-loop ligangated ion channel called the glutamate-gated chloride channel. And for short, the glutamate-gated chloride channel is abbreviated to GLU for glutamate, and then Cl for chloride. Okay, so this stands for the glutamate-gated, so the GLU is for glutamate, and I'll show you the structure of glutamate in a moment. Okay, so glutamate is going to open this channel, and then it's going to be a chloride channel. Now that's really odd. Okay, in humans, glutamate is the most important excitatory neurotransmitter in the brain, and it works through ligand-gated ion channels that are very different in structure to the cis-loop ligand-gated ion channels, okay? But the important thing is that those ligand-gated ion channels are permeable to cations. They let sodium ions into the cell and therefore cause depolarization of the electrical potential difference across the postsynaptic membrane. Okay, here in these worms we have found a cis-loop ligangated ion channel that responds to glutamate. Glutamate is its ligand, but then it's allowing chloride anions into a cell. It's going to inhibit the postsynaptic cell, basically. Okay, so this is really interesting. Right, so let me just show you firstly the structure of the neurotransmitter glutamate. Okay, so the most important excitatory neurotransmitter in the human brain is about to come up. Okay, so here's the amino group. Here's the alpha carbon. Well, I should just say glutamate is a protenergic amino acid, okay, which is actually used in proteins. Okay, so here is the core structure of an amino acid, okay, and then the R group in the case of glutamate consists of two methylene groups, which I'll just show like this, one methylene group followed by another methylene group, and then right on the end of that ethylene group you then have the carboxylic acid group, which under normal physiological pH has lost its proton and therefore the oxygen has a negative charge there. Okay, this is the molecule glutamate. Okay, and this is the strict difference between glutamate and glutamic acid, okay? Glutamic acid will be the molecule where that proton was still present on the oxygen atom there. Okay, so if that oxygen atom still had a proton attached to it, that molecule is what is meant by glutamic acid. Glutamate is the conjugate base of glutamic acid. Once glutamic acid has lost its proton, okay, and that oxygen then has a negative charge, that's what's called glutamate. Okay, it's no longer an acid note. Well, it does have the carboxylic acid group here, but this carboxylic acid group here, which has lost its proton, this is no longer an acid. It's not capable of donating protons away into solution anymore, therefore it does not fulfill the definition of an acid anymore. Instead, it's actually a base, because it can accept a proton from free solution onto that negatively charged oxygen. Okay, and that's the definition of a base, and that's why glutamate is called the conjugate base of glutamic acid. Okay, right, so I'll repeat again that under normal physiological circumstances, under normal physiological pH, glutamate has lost its proton, it's donated it away into free solution, and therefore you would have a negative charge on that oxygen there. Okay, right, so basically this can then bind to these glutamate-gated chloride channels, okay, on cells, specifically on neurons, and very, very importantly on myocytes, okay, so on muscle cells in these worms, and it can cause inhibitory currents to go into those cells. Okay, so it can make it less likely that neurons and myocytes fire action potentials, and myocytes is where we're going to be really interested in, okay, because if we could activate this channel, we would inhibit the myocytes within the worm, we'd stop the myocytes from being able to contract, and therefore we'd be paralyzing the worm, basically. Okay, and that's exactly what a drug later on that I'm going to mention called Ivermectin does. Okay, right. The other thing I want to mention now about GluCl is that it is actually just one channel. Okay, so I've just been blabbering on about how the nicotinic acetylcholine receptors, the GABA A receptors, the glycine receptors, and the 5-HT3 receptors, they are families of channels, not just one channel. Well, GluCl is just one channel. Okay, it's a pentamer of 5 identical subunits joined together, and those subunits are called the GluCl channel subunits, and you join 5 of them together and then you've got the GluCl channel, and that's that. Very simple, basically. Okay, and the next 2 channels that I'm about to show you from prokaryotes, Eunuch and Glick, they are also just one channel. Okay, right. So have I written this down anywhere? Oh yes, of course, it is a chloride channel, so that's how it can deliver a inhibitory input to neurons and myocytes. It allows chloride anions into the cell and therefore inhibits that postsynaptic cell. Okay, right. So we're now going to go even further away from humans in life forms. Okay, so we were on worms, which at least are multicellular organisms. We're now going to go to unicellular organisms, prokaryotes. Okay, bacteria, basically. We're going to see examples of cis-loop ligand-gated ion channels from bacteria. Okay, so there are 2 incredibly heavily studied channels from prokaryotes. Okay, and these have given us great insight into how the cis-loop ligand-gated ion channels in humans work. Okay, and these are called Eunuch and Glick. Okay, so they have been incredibly intensively studied. Okay, because of how similar they are to the human ones. Okay, and they are incredibly similar. That's what's incredible about this, that these channels found in these incredibly simple forms of life are the same, pretty much in structure to the ones that we have in us. Okay, they have very different sequences of amino acids, absolutely incredibly different sequences of amino acids. So the primary structure is very dissimilar, but the actual way that these proteins fold up is very, very similar. Okay, obviously they bind to different ligands, but that's because they have different primary structures. Okay, right. So let's discuss these channels then. So we'll start with Eunuch, okay, and then we'll move on to Glick. Okay, so firstly let me tell you which bacterium Eunuch was actually found in, okay? So it was found in a bacterial species called Erwinia chrysanthemi, okay? And don't feel guilty if you have never heard of Erwinia chrysanthemi. It is not one that causes disease in humans, and therefore it's not particularly notable, okay? It does cause disease in plants, so it is a problem for humans because it causes several diseases in plants that then affect crops that we want to grow, okay? So let me just draw you a little picture of Erwinia chrysanthemi then. So it's a gram-negative bacillus, okay? So what that means is that it's rod-shaped like so, and if you gram stain it, it will show up red. So let me just show this here. Okay, so this is the bacterium in which we find this channel, Eunuch. Okay, so it's a gram-negative bacillus. It'll look something like that if you gram stain it. Okay, right. So Eunuch, this cis-loop ligand-gated ion channel, again, it's just one channel. It's not a family of channels. Okay, so an Eunuch channel is a pentamer of 5 Eunuch channel subunits joined together, okay, and that forms you this ligand-gated ion channel, which will be in the cell membrane of the Erwinia chrysanthemi. So for those of you who know something about gram-negative bacteria, you'll know that gram-negative bacteria all have 2 membranes. They have an inner membrane and an outer membrane. Okay, the inner membrane is the equivalent one to the cell membrane, and Eunuch channels are found in that inner cell membrane, basically, of the Erwinia chrysanthemi. Okay, so what does Eunuch actually stand for then? Well, it stands for Erwinia ligand-gated ion channel, and for some reason they dropped the G from the name. Okay, so the E stands for Erwinia, okay, the L then stands for ligand-gated, okay, and then the IC for ion channel. So a very simple name. Okay, so what is this ion channel gated by, and then once it's opened, what does it actually conduct? Okay, well, this is again another surprise. We've just seen the Glick channel, which was this channel which was gated by glutamate, but which allowed in this inhibitory current of negatively charged chloride anions. Okay, very different from what we have in humans, where glutamate is the main excitatory neurotransmitter in the brain. Well, now in Eunuch Channel, we are going to see an example where GABA is turned upside down, basically. Okay, so GABA is in fact the ligand for the Eunuch Channel in Eunuch Channel. Okay, and we've just said that GABA, acting on GABA A receptors in humans, is the main inhibitory neurotransmitter in the brain. It opens chloride channels and therefore makes it less likely that the cell is going to fire an action potential. Okay, GABA on Eunuch Channel, acting on these Eunuch channels, is actually going to result in the opening of a sodium and potassium conductive channel. Okay, so a cation channel, basically. Okay, so again, it's completely the opposite to what GABA does in humans, basically. So, when GABA binds to Eunuch, then it's going to open this channel, which is then going to allow positively charged ions to enter the cell, sodium ions to enter the cell, okay, causing a depolarization of the electrical potential difference across the inner membrane of the Eunuch Channel. Okay, so that's a very notable example of a cis-loop ligand-gated ion channel that is present in prokaryotes, basically. Okay, and it's notable firstly because of how influential it has been on the cis-loop ligand-gated ion channel field and how useful it's been for working out how the human cis-loop ligand-gated ion channels work. And it's also notable because it proves a very incredible point that basically even these very distant life forms have very similar structural biological machines basically inside them. Okay, right. So now let's talk about the other example, which is the Glick channel. Okay, so next up we'll talk about Glick. So let's firstly talk about which bacterial species Glick was found in then. Okay, so another long name then. Okay, so Glick was found in the bacterium Glioblacter violaceus. Okay, so Glioblacter violaceus, and this one doesn't cause disease in humans either. It doesn't even cause disease in plants. Okay, violaceus. Okay, Glioblacter violaceus is actually what is known as a cyanobacteria. Okay, now cyanobacteria are bacteria which get their energy for life from photosynthesis. Okay, so they are often called algae, although they shouldn't really be called algae, because algae really should be referred to, should be refrained to being used for eukaryotic cells, which are monocellular, unicellular, and which photosynthesize to get their energy. But they are a very similar concept to algae, basically. They are bacteria which get their energy for life from photosynthesis. Okay, right. And basically, in these Glioblacter violaceus bacteria, we have found a cis-loop ligand-gated ion channel, basically, that is gated by protons. Okay, so Glick is gated by protons. Oh, and first I should probably just tell you what Glick stands for. So it stands for Glioblacter ligand-gated ion channel. Okay, so it's gated by protons this time. So if you decrease the pH around the Glioblacter violaceus, i.e. you increase the proton concentration, remember the pH and proton concentration are related in that negative logarithmic way. So when you increase proton concentration, you decrease pH. So if you decrease pH around the Glioblacter violaceus, then you get protons binding to this Glick channel, and that causes the Glick channel to open. And again, the Glick channel is just a single channel, basically. It's not a family of channels. It's one of these cis-loop ligand-gated ion channels. It's a pentamer. It's made up of 5 subunits, and those subunits are all identical, called the Glick channel subunits. Okay, protons bind to the Glick channel and then cause it to open, basically. And once it opens, again, it is a sodium and potassium conductive channel. Okay, so it will allow sodium ions into the cytoplasm of the cell and therefore will cause depolarization of the electrical potential difference across the Gloriobacter violaceus cell membrane, basically. Okay, right. So that now concludes our discussion of these basic examples of cis-loop ligand-gated ion channels. Okay, I want to stress once more that the difference between the mammalian ligand-gated ion channels that are in this cis-loop family and the non-mammalian forms, the glucial, Ehrlich, and Glick, is that these ones here, glucial, Ehrlich, and Glick, do not have that intracellular loop. So if I bring back up this picture, they do not have this intracellular loop here, the M3, M4 loop. That's the bit that's lacking. Okay, the other portions, the transmembrane domain and the extracellular domain, those are remarkably well conserved between these cis-loop ligand-gated ion channels from non-mammalian species and mammalian species. Okay, right, so we'll call it there for this video, and then in the next video what we'll move on to is discussing the structure and opening mechanism of cis-loop ligand-gated ion channels in more detail.